So hello all, good day. So we are going to work with um, the data set for so we are working on that bioinformatics. So bioinformatics, that's what you're working on. Informatics. So it's basically, in simple terms, actually, it's the application of data analysis, data science to biology. So for example, we have information or data in our biological systems. So most of these data you find it in our cells. So you talk about genomes, genetics, and so you have the chromosomes and all those data sets. So to extract the data from the cells and then to make operations or manipulations of the data, that is actually what bioinformatics is. So we are going to work with the human genome in this tutorial we are going to have now. So, but first of all, we will import the files that we need, and then I already have the data set. I have the data set and I'm going to upload it. So this is one of the main function, one of the tools you are going to make use of. Pandas is for data analysis. So we are going to import it, and then maybe we'll check the version that we have of Pandas. So this is how we check for the version. So while we are waiting for it to run, we can be uploading the data that we need. It's from my documents. Look at it, the Homo sapiens is in a zip file. So while it's running, so what is our aim or our goal in this tutorial? While we are so it has already appeared, the version of the Python we are going to use, of the Pandas we are going to use 1.3.5. It's really close. So like I said initially, this is more of like an introduction to our genomes using the Pandas and the SciFi stack for data analysis. Bioinformatics is actually an interdisciplinary field, a combination of data science and then biology, that develops methods and software tools for analyzing and understanding biological data. So it's simply like data science for biology. So in this case now, one of the data we are going to make use of, the biological data we are going to use among all the many of the biological data we have, is the genomics data. It is the most widely analyzed data, the genomes, which is what we already have now. So we are going to have access to it. I've already seen a part of the data, so we can make an explanation for the data. So for example, in the data set that we have, it contains the readme that has the file, the commenting out of the file at the beginning part of the data set. So if you're going to call the function we are going to use, we're not going to make use of those parts. Let me explain what I mean. So for example, now the columns, I already have access to the data. The columns are the column names, which is this is the sequence ID, and then this is the source. Then we also have the type, and then the start, the end, the score, the strand. phase, and the attributes. So these are the column names. So we are going to make use of this when we want to have access to this data set, but it's still loading. So if you want to read the file, you're going to use the Pandas data frame with the function read CSV, because it's in a text file, but it can also be read in CSV file because it is tab delimited data set, so it can also be read with CSV file. So write the name of the data, Homo sapiens. The 85 GFF3 GZ. So now, because it is a compressed file, you are going to use this expression. It's a compressed file. What kind of file is it? It's a gzip file, and then it's a tab delimited file, which means like let me explain what I mean by that, if I can have the access to. So for example here, no, not here. This is one of the similar files. So if you see very well in this file now, what I was trying to explain initially, has it finished loading? Okay. But anyway, so because it is tab delimited, we're going to use this argument in the file /t, because the file is delimited. And then what we are saying with this function, with this argument here, is that whenever you see any file that begins with the hashtag, we should ignore it. So that's what this thing does here. And then the low memory, don't want it to be low memory, so we put low memory. What is happening here? Low memory is opposed to false. So there is no header, so that we can make use of our column names. So there's a header is none. Now the column names should be this. It's column names. So I think this one will work. It will work. So let's run it. A very big file, so it will take a little bit of time. So it has already run. So let us see the file, the head of the data frame. Exactly. So there are 9 columns. 9 columns in this data frame. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 exactly. So this is the sequence ID. This is the source. This is the type of the genome, whether it's chromosome, biological. There are a lot of the types. So the start at the beginning of the strand and the end of the strand. The end. You have the score, the strand, the phase, then the attributes, the information that you need. So, but you see here now, there are some parts of this file that is not showing very well. So we want Python, our pandas, to display the dotted parts. So for example, you see here now, there are dotted parts here. So we want it to appear, to show very well. So what we're going to do is we come here and set our options. Pandas to set options. Display max column width, the maximum of the column width. In the old, this thing may flag an error. So in the former version of this would go with -1. So we're going to change it exactly. It was talking, we used the negative integer, so we are going to change this -1 now to none. The current one uses none. It has been deprecated. So I think this will work now. It will work now exactly. So when we run this one again now, this dotted parts will disappear. The full version will come through exactly. So exactly, have you seen it? So that's what that code does. So we check the information concerning the data sets that we have. Dataframe.info. So it's given a description of it. So for example, here you see that the start and the end are integers, and that's what's supposed to be because they are numbers, start and end. But the other ones are objects because there's a combination. For example, you see here there are combinations of numbers, of letters, and so on and so forth. So it reads it like objects. It's a mixture of integers, floats, and strings. Okay. So we want to look at the first column. Let's see what it's all about. The first column is the sequence ID. So you have df dot. You can use this to call it. You can use this way to pass the string of the column inside and use unique. It will work. It will work. Sorry, unique. The unique values of it exactly. You can also use this way to write it directly to the data frame, and it will also work. So these are the unique values of the sequence ID. So what will be the shape? It's lot. Unique. The shape of the data, this slice of data. So we have 194 rows. So there are 194 of the sequence ID. So it includes the chromosomes, which starts from 1 to 22, and then the XY, and the mitochondria, which is empty, as well as the other 169 sequence IDs. The ones that are starting with KI and GL are DNA sequences, like these ones. The KI starts with KI, these ones that start with GL. They are DNA sequences or scaffolds in the genome. And so they have not really been successfully assembled into chromosomes. They are on their way. So this is about the biological part of what we are doing. So the human genome draft, though it came out more than 15 years ago, the current human genome is still incomplete. Yeah. So the difficulty in assembling these sequences is largely due to complex repetitive regions in the genome. So let us look at the other column, the source column. So the data frame dot source. So it's going to show us the counts of each of the values in the source column. So we have the Havana ensemble, and so on and so forth. Mayberries, GRCH38. So this function, um, this function, this method actually is very useful for a quick count if we are working with categorical variables. So we just simply use the value count, and it just gives us a brief of the values of the categorical variables. So there are actually 7. 1, 2, 3, 4, 5, 6, 7. 7 possible values for this column. And where the majority are in this first 3 columns: the Havana, the ensemble, Havana, and the ensemble. So eventually you see the relationships about what sources mean and the relationships between them. So to make this thing simple, we're going to work with the first 3 values of this source. So we're going to work with the Havana, ensemble, Havana, and ensemble. So how much? That's another question. How much? So we can just change this into text. It's the text. How much? How much of the genome is incomplete? We find out from the data that we have. You can tweak this up, make it a little bit thick. How much of the genome is incomplete? So the information actually of each entire chromosome is in the entries from the source GRCH38. So we filter it out first. So we are going to work with this particular area, this source, this one. So wherever the source is equals to this, that's what we want to get out. So we are going to make an operation, a Python operation, and create a variable for that to contain our data. So we call it GDF. So if you check this data frame where the source is equals to GRCH38. So it's going to select it. So let us see the shape of that GDF.shape. So there are 194 rows and then 9 columns. Let's just look at it. So you have the GDF.sample, a sample of the data set. Wow. Okay. So this 194 is familiar. You have seen this 194 before. The length of this is actually 194 when we run the shape here. Exactly. The sequence ID 194, and we also have 194 here. If you see and figure out what is the correlation between why there is the same number of sequence IDs, also the same number of GRCH38. So okay, what it means is that if that the 194 unique values actually in the sequence column ID, so in the sequence ID there are 194 unique values. It actually corresponds to each of the entry of the GDF corresponds to one each of the 194 values of the sequence ID, and it makes sense because if you look at here now, so we run this one now, the source is, if it is 1, look at 1 here. What I mean is that, so the sequence ID is from 1, we ran it 1 to 10, 11, 12, 13, and went on to this value. If you take one of these values now, so let us say we are taking let me say 11 or maybe 12. It will have, the 12 will also have that that sequence ID, this it will have a source of GRC35. So for example, check this df where df dot source. No, this is the sequence ID. Sequence ID is equals to this value now. You watch very well, it has that GRCH38. Any number you put in here would have this GR. So if you put 13 here, it will also have it. Are you seeing that? Even if you collect any of the values here now and insert in that same area, in that same place, it will also have one line that has that GRCH38. Are you seeing that? So what this thing means is that for each of the sequence ID, there is a line that has the source for GRCH38. So there are a total of 194 sequence ID, and each of which has a corresponding GRCH38. That's the reason why the GRCH38 is also 194. That's why it's 194. And if you watch very well, these sequence IDs are all unique. So because it's one line from each of the sequence IDs, they are all unique values here. So, but if you move further in what we are doing, so we want to. So if you see that the unassembled sequences are of the type supercontig, while the others are of the chromosome. So these the ones that are of chromosomes have already been assembled. For here now, the ones with chromosomes are assembled, but the ones that are supercontig are they're not yet assembled. So we are going to compute the fraction of the genome that is incomplete. So before we do that, we are going to find out the length of the entire genome, which is the sum of the length of all sequence ID. So but let us have a copy of this, a copy of the GDF, which is equals to GDF.copy. So this copies, so you don't want to affect the original file. So that's why we have a copy of it. Now we are going to perform an operation now. So we are going to create a new column, GDF, which is length, a new column called length. So this will be, we get our the column, the end column, which is this end column. We are going to subtract it from the start column, the differences, and then we add it one so that we make up with to make it inclusive. So it gives us the length of the chromosome. So let us see the head of the data set. Exactly. So we have a new column here, the length. So now that we have the length, what is the sum of this length? So GDF dot length, the sum of the lengths. We have how many million? 1, 2, 3, 4, 5, 6, 7, 8, 9. Hey. In billions, 3.09 billions. So, but we want to select the ones that are not chromosomes, that have not been assembled. the unassembled ones. The unassembled ones are the ones that are not of chromosomes. So we are going to perform a very beautiful operation now. So for chromosomes, if you want to check for the chromosomes, this is the chromosome, which is, you want to see the string of the value. So we can use this operation in range 123 because the first part of the chromosomes are 1 to 22. You see, it's arranged in a way that, because it's in string format, so it's not arranged numerically. It's arranged in a way, in quote, alphabetically, because wherever the ones begins, these are the first ones before twos. That's why you have 1 and then have 10, because 10 starts with 1 and 11 also starts with 1. That's why this first one. So, but if you look clearly very well, you see that it starts from 1 and ends at 22. So we want to have 1 and 22 inclusive. That's why we are using 23, the range of 23. And we use this string to make the operation of what we are looking for to be actually the string, because this is in a string format. So we don't want it to be like an integer. That's why we use this. We casted it into string. So this is a list. We're also going to add it with the remaining part of the chromosome, which is X, and then Y, and then empty mitochondrion. So this is our chromosome. If you watch very well, if you look at this, you see it is going to give you the data. You see everything is in string format, not in the integer, and then our X, Y empty. Yeah. So that's it. So, but we are not looking for the chromosome, so there should be a negative operation. So we are going to check wherever this GDF. So we are looking for GDF, the sequence ID. Is it in this list that we have already created? Chromosome. Then the length of it, and then the sum of the length. That is for chromosomes, but we are not looking for chromosomes. We're looking for the ones that are not in the chromosome. So we are going to put the negative operation, which is the minus. So this is the unassembled part of the data, and then we're going to divide it with the total length of the GDF. GDF dot length dot sum. What is the percentage? 0.003. So if you calculate this, remember your mathematics. So 0.0037. If you divide it by 100, 1, 2. So we have 0.37%. Very minimal amount of unassembled data. 0.37% of the data is unassembled. We can actually perform these operations by checking the value of the type. So the type that are assembled, that are okay, are chromosomes. But the ones that are not assembled is actually supercontigue. So, like for example, here now the chromosomes, the type is chromosome, so these ones are assembled. But these ones that have the type are supercontigue. They're not. They're not assembled. So we are going to reuse that here. So GDF. Here GDF. The type of the GDF is equals to supercontigue length of sum. So you divide it by the total length of sum. GDF dot length. So it will still give us the same value. Yeah, exactly the same value. So the next one we're going to do is how many genes are actually here. How many genes are there in operation? So we want to find out. So we are working with so we are going to focus basically on the source, the ensemble, the Havana, and the ensemble Havana. This is where the majority of the annotation entries belong, and the larger share of the national entries. So we're going to create a new variable. Data frame, where the data frame dot source is in ensemble. And then Havana. Ensemble Havana. So let us see a sample of the data. This is a sample of the data. Okay. So there are a lot of types here, mixed up types. In this ensemble Havana. I don't know whether we even have chromosomes here. So we are going to have the count of these types. So we're going to use the value counts for the type. So we have EDF dot the type column. You should count the value count of this to give us a brief description of the types that we have here. Wow. So the highest three here are the exome CDS and 5' UTR, followed by 3' UTR. These are the largest. The exome, so the exome, the coding sequence is CDS, and then the untranslated region, UTR. So these are subgene elements. We are looking for gene count actually. As shown, there are around how many of these here? The gene count. This is what we are looking for, and from what we see here, it's actually 42,470. So what are their names and what do they really do? To answer this question, we need to look closely at the information, the attributes. So we need to select this gene wherever the type is gene. So we have this. Where the type is gene. And then we have a copy of it like how we did before, so that we will not tamper with the main data set. So we're going to look for the column. The attributes column. And then see the values that are in that column. So these are the values of the attribute columns. So you're going to perform like a regular expression. You have to extract this because these are like dense kind of information. So for example, if you want to access just one of it, so one of it, just one, you see that there are a lot of information that are hidden here. So you even want to find, to extract this gene ID, to extract this gene name, and maybe the description. So but we are going to see how we are going to do that. So they are really formatted as semicolon, separated list of tag value pairs. So the information we are most interested here from this data is the gene name, the gene ID, and description, and we extract them with the regular expression. So we are going to use the module, the RE module for regular expression. We are going to import it. Yes. No, this. So the gene name. So re.compile. So what we want to get is wherever we have this kind of information, the name, as we can see here. So for example here, what's similar here? Name, for example here, is equals to this value now, for example. So we're going to say name is equal to. How can we find what we need? So we are going to start with the operation. This operation, we'll explain this later. And then, so the gene name. And the plus question mark. And then the end here. And then semicolon. I will explain what I'm doing. So we have the extract gene name, attributes, string, is supposed to re gene name search. Yes, that'll be this. Then we're going to return so from here now, what we're trying to do now is, so you see, we used plus and the question mark instead of just not ordinarily plus here, because we want it to be non-greedy. There's something called non-greedy, so that it should not continue after it has already found what we want it, and then the semicolon. It will continue to move again to find another one with the semicolon. But once you put this plus and question mark, it stops there once it finds what we have already wanted it to find. And then this other part of so you notice very well here that we first of all compiled it before we now search it. So it's done. It actually is for better performance because you apply it to thousands of attribute strings. So we first of all compile it and then we now search it. So this will be a helper function. This extract gene name. The helper function that we are going to. So you have helper functions. Helper functions are the functions that we can integrate into our data frame. I will show it in an example. That we perform the operations inside the data. So you have a lot of operations you want to perform in the columns, for example, in a specific column. So for each of the rows of the columns, we want to perform these specific operations in it. So we use the helper function, which is inserted into a method .apply that is being attached to the data frame or the slice of the data frame. So this is the helper function we're going to do. It's going to perform these operations which are inside here in all of the rows. These are rows of this slice of this data frame. Now, so this is the helper function. So you now create a new column here, which is called the gene name, because that's what you are extracting. So we have the attributes column, which are going to perform the operations on attributes. So we use the apply function that will take in the helper function. Exactly. So it will work. So if you watch very well now, if you do this ndf dot head, you have a new column that has been extracted, which is called the gene name. Look at the column here, the gene name. This ddx11l1 was extracted from here, just as how this wash washZP was also extracted from here. So that function, this function we have here, this extract gene name, was applied to each of the row of this particular slice of this data frame ndf data attributes to give us this eventually. So we are going to do that we have already done that to get the gene name. We are still going to do that to get the gene ID and then the description, but that will be in the next part of this tutorial. But for now, this is like a glance of what we can get in analysis of biological data using Pandas and Sci-Fi. Thank you very much. In the next video, we are going to continue with the part of the gene ID and then the description. Okay.